Okay, so the four classes of antivirals we'll be covering today is exit inhibitors, entry inhibitors, viral replication inhibitors, and finally RNA I. Let's begin with RNA I. RNA I is a short RNA sequence of about 20 to 25 bp length, and it works by interfering with a selected RNA string, and the way it does this is by having what's known as a complementary sequence to the RNA. It will bind itself to the RNA, thereby rendering the RNA useless. So what does complementary sequence mean? Yes, it simply means that the RNA will have an A where there is a U and a C where there is a G. So basically it's an inverse copy of the RNA it targets. And the mechanism of action for any RNAi-based drug is that it will 1. Bind to the RNA, 2. Alter its structure, and finally cause the ribosome to reject the transcription. Exit inhibitors works exactly like they sound. They make the virus stuck in the cellular jail. But before I'm going to explain any further, it's important to understand just how rude of a houseguest a virus is. Because what it does is that it says thanks to your cells for utilizing their energy by simply cutting a big hole in them. At least some viruses does this. And the weird crown-shaped one is definitely one of them. But anyways, how do the exit inhibitors works? You see, viruses utilize something known as proteases to escape a cell. Wow, hold on dude, what is a protease? Allow me to explain. A protease is simply an enzyme that cuts a protein. So basically I've seen like two mechanisms of action for these antivirals. One is simply to inhibit the protease, i.e. they fooled the enzyme to cut them instead of the protein, and after the cutting there is an atom like either a chlorine, fluoride, oxygen, or nitrogen, which then breaks off the inhibitor molecule and leaves the inhibitor covalently and permanent bound to the enzyme's active site. 2 is that they become a drug which causes the cell to commit apoptosis when they get in touch with that viral protease. Apoptosis just means cellular suicide, by the way. Viral replication inhibitors works by simply disrupting the viral division/replication machinery. And the way they do this is by looking like a nucleobase, one of the 4 bases of RNA or DNA. And then when the enzyme that replicates the virus tries to utilize them in the fabrication of the viral genome, it goes haha, I've got you now, and then it permanently binds the active site again by having an electronegative atom break off with a hydrogen atom from the active site. And this in turn causes the active site to covalently bind the atom on which the electronegative atom sat. Entry inhibitors works exactly like they sound, like they either bind the viral capsule protein or the receptor which the virus uses to gain entry into the cell. Now if they bind the receptor, they are most likely antagonists of that receptor. Antagonist just means a drug which blocks the active site of a receptor by binding slightly outside of the active site on the receptor protein. Or if they bind the viral capsule, they change its surface chemistry so that it becomes repulsive towards the receptor instead of attractive. Or simply put, they change the viral capsule protein's electric field so that it becomes repulsive towards the receptor it targets, or they simply make the capsule protein too big to fit in the active site. So that's it folks, see you in the next video. Please like, share and subscribe.